Welcome to the Deep Dive, where we turn complex research into critical knowledge. Today, we're plunging into the world of the immune system, looking at a cell that's, well, often misunderstood, the macrophage. Think of it as the body's ultimate cleanup crew.
That's a great way to put it. They're absolutely essential for keeping our tissues healthy, a state we call homeostasis. And a huge part of that job is something called efferocytosis.
Efferocytosis, that's basically just eating dying cells, right?
Exactly. It's the process of engulfing and clearing cells that are programmed to die apoptotic cells. They have to do it fast. If they don't, those dying cells can burst, spill their guts, and cause some really damaging inflammation.
Right. And for the longest time, the thinking was that all dying cells were, basically the same. Once a cell started dying, it just put out this universal eat me flag, and the macrophage saw it as, well, trash to be collected. Simple as that.
It was a simple model, but it turns out it was a bit too simple. We've always known that what a macrophage does is heavily dictated by its local environment. It's always listening for signals.
And one of the loudest signals for time to repair is interleukin-4, or IL-4.
Yes, IL-4 is the classic type 2 cytokine. It essentially tells the macrophage, okay, cleanup is done, now it's time to rebuild.
So here's the gap, the big question this new research gets at. We know the environment, the IL-4 matters. We know the cleanup matters. But does the identity of the trash itself, does the specific type of dying cell change how the macrophage rebuilds?
Precisely. Does the meal change the chef's specialty? That's the question.
I love that. So in this deep dive, we're going to unpack the new evidence showing that the apoptotic meal is, in fact, a critical trigger for macrophage function. We'll look at lab results, a really fascinating parasite model, and the specific receptors that make it all happen.
Yeah.
Okay, so let's get into the experiment itself. How did they set this up in the lab?
So they started with bone marrow-derived macrophages, BMDMs for short. They're sort of the perfect blank slate macrophage for this kind of work.
And what was on the menu you mentioned?
Well, they offered them three very distinct types of apoptotic cells, or ACs. First, you had apoptotic neutrophils. These are your immune system's first responders.
Okay, the foot soldiers.
Exactly. Then they used apoptotic thymocytes, which are a type of developing T cell. And third, apoptotic hepatocytes, so liver cells, a totally different non-immune lineage.
So you have an immune responder, an immune trainee, and a civilian tissue cell.
Perfect way to put it. And the key was they fed these cells to the macrophages while also stimulating them with that IL-4 repair signal.
And by looking at all the active genes, the transcriptome, they saw what, 3 different outcomes?
3 completely distinct functional signatures. It was remarkable. When the macrophages ate the apoptotic neutrophils, the AN cells, they switched into what the researchers called a tissue remodeling or wound healing profile.
The full construction crew mode.
Absolutely. We saw huge increases in classic repair genes, things like ARG1, retinol, which makes a protein called REL-M-alpha, and CHIL3 or YM1. These aren't just letters. I mean, ARG1 literally provides the raw materials, the building blocks for making new collagen and rebuilding tissue.
Wow. And this wasn't just a blip on a genetic readout.
No, they confirmed it at the protein level. The macrophages were pumping out RLM-alpha and YM1, but only when they ate neutrophils, not the others.
So what happened when they ate the liver cells, the apoptotic hepatocytes?
The complete opposite. Instead of a repair signature, they got an immunosuppressive or tolerogenic phenotype.
Tolerogenic, so stand down, don't react.
Exactly. Tolerate this, suppress the local immune response. They saw genes like SOX1 go up and, really critically, CD274. CD274 is the gene that codes for PD-L1.
Wait, PD-L1, that's the immune checkpoint, the thing cancer cells use to turn off the immune system.
That's the one. So eating a dying liver cell seems to tell the macrophage to actively shut down local immunity.
That is a wild difference. Aggressive repair versus active suppression. And the T cells, the thymocytes.
They were kind of the middle ground. They didn't really push the macrophage strongly in either direction. It was really the neutrophil and the hepatocyte that created these two totally divergent paths.
And it wasn't about, say, how much they were eating.
Great question. And no, they checked. The macrophages ate about the same amount of all 3 cell types. And even when they tried to force feed them more of the other cells, they still couldn't trigger that powerful tissue remodeling response.
So it's not the quantity. It is, without a doubt, the quality, the identity of the meal. Okay, let's just pause and unpack that for a second. This completely changes how we should think about tissue healing. Before, it was just clean up the trash. Now we see that the type of trash determines whether the kitchen, starts cooking a 5-course meal or just shuts down for the night.
It really does.
Yeah.
And if you think about it, this starts to explain why macrophages are so different in different organs. Macrophages in the liver are known to be very tolerant, right? Well, maybe it's because they're constantly eating dying liver cells.
Ah, so they're being continuously programmed for tolerance by their local diet.
Whereas in a tissue that's constantly being repaired, maybe they're always munching on neutrophils and are just primed for remodeling.
Yeah.
It's programmed by the local cell turnover.
Which leads to the billion-dollar question, how? What's the signal? If that universal eat me signal, phosphatidylserine or PTD-SIR, is on all of them, what makes the neutrophil so special?
So the researchers went into full detective mode here. First, they crossed PTD-SIR off the list. They confirmed all 3 cell types had similar levels of it on their surface.
Okay.
But here's the killer experiment. They made these little bubbles of fat liposomes and just coated them with PTD-SIR. So the eat me signal with no cell inside.
The wrapper without the candy?
Exactly. And when they fed those to the macrophages, nothing. It wasn't enough to trigger that remodeling gene signature. So PTD-SIR gets the macrophage to eat, but it's not the signal that tells it what to do after eating.
So it's not an external signal. What about a secreted one? Did the macrophage eat the neutrophil and then, send out a memo to other cells?
They tested that too with a conditioned media experiment. They took the liquid that the neutrophil-eating macrophages were sitting in and put it on fresh macrophages. Nothing. The signal didn't transfer. It confirms the effect is all about direct cell-to-cell contact and, crucially, what's inside the cell that gets eaten.
The Trojan horse idea. The real message is hidden inside. So what did they find in there?
Lipids. It came down to the fat profile. They did a deep lipidomics analysis, and it turns out the apoptotic neutrophils were just stuffed with certain kinds of fats compared to the liver cells, specifically free fatty acids, or FFAs, and a couple of others like PEs and sphingomyelins.
So the neutrophils are a much richer, fattier meal.
In a very specific way, yes. And when the macrophages ate them, they started accumulating these FFAs themselves.
And then they turned on genes that are controlled by these internal sensors called nuclear receptors, things like PPAR gamma and LXR.
So the lipids from the dead neutrophil literally go inside the macrophage, flip a switch on these receptors, and rewrite the cell's software from the inside out.
That's the mechanism. The meal itself contains the instructions.
That is an incredible finding in a petri dish. But the real test is always, does this actually work in a real, complex, living animal?
Right. And for that, they turn to a mouse model of infection with a parasitic worm, Schistosoma mansoni.
Schistosomiasis. Why that model specifically?
It's kind of the perfect storm for this question. It causes this chronic liver damage with lots of inflammation and fibrosis, and you get a ton of dying cells, neutrophils, T cells, liver cells, all of them. And most importantly, it's a very strong type 2 immune environment. It's swimming in that IL-4 signal.
So it's the exact environment they modeled in the dish, but, messy in a real liver.
Exactly. So they took their specially programmed macrophages, the ones they'd fed apoptotic neutrophils and IL-4 in the lab.
The MO+AN cell.
Right. They took those and injected them back into the infected mice. And the result was pretty stunning. These macrophages dramatically decreased the number of parasite eggs stuck in the colon. They were actively helping to clear the infection and reduce the disease.
Wow. So this isn't just a biological curiosity. This specific macrophage phenotype, it has a real protective therapeutic effect in vivo.
A profound one.
But how? How did they help clear the eggs? What was the proposed mechanism?
This was a surprising twist. It seems to come down to blood clotting or coagulation. Those MO+AN macrophages were loaded with genes for coagulation factors like factor 3, factor 7, and factor X.
So they were promoting clotting. That sounds counterintuitive.
It does, but the theory is that they're helping to form these stable, controlled clots around the eggs as they move through the tissue. It's like they're building a biological barrier, a scaffold that helps contain the eggs and guide them out of the body through the intestine instead of letting them get stuck in the liver where they'd cause fibrosis.
So it's not about attacking, it's about infrastructure management, building roadblocks to safely escort the problem out of the building.
That's a great way to think about it.
This really ties it all together. It's not a lab trick. This specialized population of macrophages can actually improve disease in a living animal. And we've ruled out the simple external signals. We've pointed the finger at the lipids inside.
But the big question still hanging out there is, how does the macrophage pick its meal? In that chaotic, inflamed liver with all sorts of dying cells around, how does it selectively find and eat the apoptotic neutrophil to get those instructions?
The message is inside, but you need a special key to get through the front door.
Exactly. And that brings us to the receptors on the macrophage surface. specifically two called AXL and MERTK.
AXL and MERTK, I've heard of them. They're known to be involved in clearing apoptotic cells. But are they generalists or are they specialists?
Well, this study argues very strongly that they are specialists. The researchers created macrophages that were missing both AXL and MERTK, a double knockout, and they ran the feeding experiment again.
Okay, and what happened when these receptor-deficient macrophages tried to eat the apoptotic neutrophils?
They couldn't do it very well. Their ability to eat the neutrophils was significantly impaired.
And for the other cell types?
And here's the punchline. Their ability to eat apoptotic T cells was only a little bit reduced, and their ability to eat the apoptotic liver cells was not reduced at all.
Whoa. So AXL and MERTK are the VIP entrants, specifically for apoptotic neutrophils.
That's the breakthrough. The apoptotic hepatocyte uses a different door, a general admission ticket, but the neutrophil has the key for the AXL-MERTK lock. These receptors aren't redundant. They are a critical switch for determining which meal gets prioritized and, therefore, which program gets turned on.
So what happened when they did this in the mice? When they knocked out AXL and MERTK just in the macrophages of the infected mice?
The disease got much, much worse. It was a dramatic effect. The mice couldn't clear the eggs properly. Their liver damage markers, ALT, went through the roof, and they had a massive accumulation of collagen, which is, the hallmark of fibrosis, scar tissue.
Because they couldn't get the right meal. They couldn't eat the neutrophils, so they never got the signal to turn on the repair and clearance program.
Precisely. The whole downstream effect was blocked. You didn't get the upregulation of those key remodeling genes like retinol and CHIL3. This really connects all the dots. It's AXL and MERTK-dependent eating of neutrophils that controls the whole beneficial response.
So let's try to synthesize all of this.
Yeah.
It's not enough to just have a macrophage. It's a system that needs three things to align perfectly. You need the right environment, the recipe, which is the IL-4.
Right.
You need the right ingredient, the specific identity of the apoptotic neutrophil. And you need the right tool, the specific utensil, which is the AXL-MERTK receptor, to bring that ingredient in. Only then do you get the right dish. Tissue repair.
It's the perfect summary. And this opens the door to thinking about therapeutics in a totally new way. We could call it selective macrophage feeding. Instead of just broadly trying to reduce inflammation, what if we could design therapies for diseases like liver cirrhosis that specifically trick macrophages into eating the right kind of meal?
Forcing them into that pro-repair mode.
Exactly. You could commit them to the function you want, regardless of the messy environment they're in.
It's just a fundamental shift in our understanding. The idea that the identity of a dead cell determines whether the outcome is repair or tolerance is...
It's huge.
It is. It tells us that a macrophage's destiny is written by the death that surrounds it. And here's a final thought for you to chew on. If AXL and MERTK need a specific key on the neutrophil surface to open, what if we could figure out exactly what that key is?
And if we had built it ourselves? Could we create a tiny synthetic particle, like a nanoparticle, that's decorated with that specific key, a synthetic apoptotic neutrophil? You could then inject this particle, have it go anywhere in the body, and selectively tell macrophages, start the repair program now. You could potentially reverse fibrosis anywhere. That's the future this work points to.
A custom-built microscopic repair signal delivered right where it's needed. A fascinating place to end. Thank you for diving deep with us.